I would like to welcome Dr. Devinder Sehgal to chair the preceding session. I also request on dais Dr. Aashini Parikh, who is currently working as Deputy General Manager in Clone Development and Immunoassay Development and Immunoassay Team of Biotechnology Department, Zydus Research Center, Ahmedabad. I would request Dr. Sehgal to introduce the speaker. Thank you.
So we will have Dr. Aashini Parikh giving us this lecture on making of monoclonal antibodies. So she is a principal investigator who is well experienced in biosimilars and novel biologics. She has a great background in cell and immunoassays, clinical development. And also, she has been with Zydus Research Center for many years now. So she is involved in developing assays for pharmacokinetics, pharmacodynamics, and immunogenicity assessment, which are crucial for preclinical studies and clinical development. So I will hand over the podium to Dr. Parikh, please.
Thank you, sir. So, good afternoon, everyone. And this presentation is going to be a bit different from what you have heard. Till now, you have been hearing about the immunology basics. Now, I'll be trying to give you an insight into how you're going to develop an antibody, a monoclonal antibody, as a therapeutic. So there are some aspects which already many of our respected senior speakers have already taken you through. I would also like to again take you through the market of biologics. This picture actually tells you these are the top 10 best-selling therapeutics of 2023. And interestingly, if you see, 5 of them are monoclonal antibodies. This itself tells the importance of this space in the therapeutic area. Just to bring to your notice, more than, I think, around 180 monoclonal antibodies have been approved till now, world over. And there are at least 570 therapeutic mAbs which have been or are being tested in different clinical trials. So again, we have discussed about the global landscape, but I just again wanted to bring to your notice the recent 2023 pie chart, where you again see that monoclonal antibodies make up 71% of the biological development. And there are 2 additional classes of monoclonal antibodies, ADCs and bispecifics, again coming up in a big manner. They have their own advantages. I'll be touching base upon them in my subsequent slides. And then there are other fusion proteins which we have already talked about, fragments that can be just FC, Fab fragments, and co-formulated therapeutics. In addition to that, again, we saw that cancer is an area where you have maximum monoclonal antibodies being developed, followed by immune-related diseases, and then the infectious diseases. The right-hand side, This right-hand side chart, which I wish to show, was the aim for showing this chart was that there are different targets being tested in different clinical trials in cancer as well as infection disorders. But PD-1 class of molecules has been taking over the space in quite a large manner. Going back to what I earlier shown, here Keytruda and Opdivo are the 2 molecules which you see are anti-PD-1 molecules. And why they would be having a large market is because they would be catering to a number of different cancers because they are targeting T-cell immunity. So it is not a cancer target which is being targeted, which would be specific for that particular cancer, but it has the ability to cater to or treat many different kinds of cancers. Again, the developmental journey of therapeutic antibodies have been shown by the previous speakers. So again, I'll not go much into detail. But again, to highlight that we have been working on therapeutic antibody generation since a long time now, starting from hybridoma to Fach display to transgenic animals, yeast display, all these things have been discussed. The first map was a murine monoclonal antibody, which was approved way back in 1986, followed by the first ADC or the antibody drug conjugate approval, which is now one of the important classes of monoclonal antibodies in the current space. And fully human antibody, which has its own advantages, which have been again discussed by the previous speakers. A bispecific antibody being approved in 2009. And I'm highlighting the anti-PD-1s which have been approved recently because they have been quite effective in treating many different cancers. I'll just skip this slide. Now, just coming to the definition, we all know what are monoclonal antibodies. So they are produced by a group of antibodies. They are a group of antibodies which are actually produced by identical clones of B lymphocytes against a particular antigen, and they are similar in the terms that they have they would be same in the protein sequence. They would be binding to the same antigen site and have same binding affinity to the target, and also carry out similar downstream functional effects, the ADCC and CDC effector functions which we talked about. I'll not go to, in the details of this antibody structure and functions that has been already explained to you. I'll just touch base upon the mode of actions for the therapeutic antibodies. So an antibody can bind to a target which can be soluble or expressed on the cell. And it can be just a blocking antibody that it is not allowing its target to bind to some other receptor and carry out its function. So here, in this case, you might not require effector function. It is just a blocking mechanism that you are trying to have. The other would be where immune effector functions are playing a role. So for example, a target is being expressed on cancer, and there your antibody goes and binds, and then you have this effector functions like ADCC, ADCP, and CDC, which would aid in killing your cancer cell. The third would be signaling antibody can be agonist in nature, that it can help the proliferation of a particular cell of interest. It can be T cells. You can have activator molecules which you want to further activate. And then you have targeting antibody where you have ADCs or the antibody drug conjugate, which I just spoke in my earlier slide. So what this antibody has is it is fused, or it is tagged to a chemotherapeutic agent. So the antibody just brings the antibody drug conjugate would go and bind to the target present on the cancer cell, and then it will help release this toxin within the cells and in the surrounding, helping to kill the cells. There were various properties of antibodies which were talked about by earlier speakers, but I think this no one touched, so I just thought of touching it. Monoclonal antibodies are expected to have higher half-lives, as we all understood from our previous talks. So why does a monoclonal antibody or antibody-like molecules have a longer half-life? So what happens is that there are FcRn receptors, or they are called neonatal FcRn receptors present on the surface of endothelial cells. The antigen will bind to the cell and will be taken up in a form of endosome. And within the lysosome, this the antigen or the other proteins which are bound to it can separate out. But this receptor, FcRn receptor-bound monoclonal antibody, gets recycled outside the cell while it is still bound to the receptor. So binding occurs at an acidic pH while its recirculation occurs in a basic pH. This helps the antibody to keep on recirculating in the sera, and hence it is expected to have a larger half-life, and hence you'll have monoclonal antibodies dosing which are done mostly they are either bi-weekly or every 3 weeks, because the half-life of monoclonal antibodies is around 21 days. I'll not again go into these formats. They have already been discussed by other speakers. This also we have understood that earlier murine antibodies were being made, and now gradually we have come to fully human antibodies. And the advantage of having fully human antibodies is that they are expected to have very less immunogenicity when given as biotherapeutic drugs. These are the suffixes which are used with the different kinds of antibody. Like for example, with humanized antibody, you use Zumab. Umab is used with a human antibody. Cmab is used with a chimeric antibody. Again, this slide has been shown by a previous speaker. I'm not going into those details. Coming to the antibody-drug conjugates. Now, what are antibody-drug conjugates? So basically, it's a monoclonal antibody which is covalently linked to a cytotoxic drug, which we call as payload, via a linker. So this molecule is what we call as antibody drug conjugate, and it's very complex molecule. There are a number of payloads which are available, or the toxic drugs which are available, which you can use to use. And also there are different kinds of linkers available, which can get your payload bound to the antibody. Now what happens is, when there is an antibody drug conjugate, when it binds to the target present on the cell, it gets internalized. So internalization of that particular antibody and target is essential to make an antibody drug conjugate out of it, and so that it works. So when it gets internalized, the payload gets detached from the antibody. The antibody gets degraded. By this payload, it's being a chemotherapeutic agent itself, causes killing of the cell, and as well as it can go out of the cell and kill the surrounding cells. So it has dual function. This is called as a bystander effect. So it can kill the cell itself, a tumor cell in which it enters, and the chemotherapeutic drug can kill the surrounding cells also. But it has its own disadvantages, like it can also cause non-specificity and killing of some normal cells in the surrounding area as well. So it has to be a very good balance of what linker you are choosing, what payload you are choosing to tag the antibodies and make them effective. 15 ADCs have been currently approved in various countries, and a number of clinical trials are ongoing. And they are against various targets, as you can see in the lists. And there are various linkers, various payloads which are being tested. DAR is called drug-antibody ratio. Usually 3 to 5 is the ratio which people tend to keep to have a balance of safety and efficacy. So I'm not going into details of all these things. Bispecific antibodies, again, which we haven't touched or discussed in the past lectures, so I'll just touch base upon them. So we know from the name itself what are bispecific antibodies. So bispecific antibodies basically have, they are antibodies which can bind to 2 different targets. And what is the advantage of that? One is that we have been talking about, and I think someone in the audience asked the question that why sometimes resistance is created and very effective cancer molecules do not work like EGFRs. So sometimes what happens is that the cancer, the target or the receptor which a cancer is expressing, it either gets mutated or the body or the tumor stop expressing that particular antigen of interest as a response to survive. So tumor wants to survive, and in response to that, it is trying to downregulate that receptor expression. So in that case, if you have 2 different antigens being targeted on a cancer cell, it will help you to come out of such situation. Secondly, the role of the bispecifics is also like it can bring 2 cells together. For example, this is an example which is a CD3 and CD20 bispecific antibody. So what will help it doing is it will bring the tumor and T cell in a close proximity so that it can help in killing the tumor cell. Or it can bind to 2 different targets and which are present on the same or different cells, and prevents its interaction, leading to, again, whatever mechanism you want, in this case, prevent clotting. There are many different formats of bispecific antibodies which we have discussed, and I am not going into details of those things. There are at least 13 different bispecific antibodies which have been approved in different indications and different lines of treatment, say if there are 2 lines failed, 3 lines failed treatment, etc. The chimeric antigen receptor, again, is something what we actually some of the speakers did talk about it in brief, but this is just one slide showing like what is chimeric antigen receptor. So chimeric antigen receptors, T cells are, what they do is, T cells are taken from the patient and they are modified to express an SCFE of interest, which is against the target present on the tumor cells. So you have lentivirus or retroviruses being used for that purpose. So you isolate the T cells, you put in your gene of interest into it or vector of interest, and then return back those CD4, CD8 T cells into the human being. So what it does is, like, this is the structure of a CAR T cell, which has SCFE, and it has the downstream signaling domains which lie inside the membrane, and it interacts with the tumor cell, and there is a downstream signaling which happens, which leads to activation of T cells and also proliferation, leading to tumor cell death. There are there's a typo error here, I'm sorry for it. There are around 8 different CAR Ts which have been either approved or in the approval stage. The pink ones are from India, which have been developed using indigenous technologies. Immunact is the first CAR T antibody which has been approved. Immunil also has a CAR T antibody where it is under approval, and I guess in a short time it will be approved. So this is one more therapy which is coming up, and it has shown very good responses, very good complete responses, and so it is good that India is moving towards developing its own CAR T therapies. So now this was sort of an introduction of different kinds of antibodies and the market, etc. Now how we discover and then develop a recombinant antibody so that it is used as a therapeutic. One thing, I'm not sure whether the students are aware about it or not, but there are 2 types of biologics which we develop. One is a biosimilar molecule, which is sort of a generic molecule, what we call like paracetamols or such class. So generic word is used for small molecules, a biosimilar word is used for larger molecules. The reason being that because biosimilars or biological products are being developed in live systems, cells, biological systems, and no two biological systems can be same. There are a number of different things which can impact the the quality of a protein being produced in a biological system. So one is a biosimilar, which actually you try to make a copy of already existing molecule, and one is a totally novel biologic which has never been tested into humans. So this is the development pathway, as you can see. In biosimilars, because its safety and efficacy has already been tested, the emphasis is on its physicochemical characterization. I'll be coming into detail in the subsequent slides, like what parameters are important and why they are important. While you need to do very low number of clinical trials, at least just one clinical trial, maybe phase 3 and one PKPD, while the scenario for developing a new biologic is totally opposite. Because it has never been tested for its safety and efficacy, you need to do a large number of clinical trials, and of course structure and function is important, but this is the most important thing. So discovering of novel therapeutic monoclonal antibodies has already been discussed. I'll not be going into the details again. So what is the flow of discovering a novel antibody? First is that you have to decide on what target you want to work on. Secondly, you have to validate that, yes, that is a correct target to work on. How do you validate? You read papers, you try to understand whether people have been working on that target or not. Then is an antibody discovery, which I showed in the previous slide, that you use one of the technologies to find out a novel molecule which is binding against your target of interest. The third is, once you have some lead candidates, you try to find out the affinity of that molecule, because it is going to be important. Higher the affinity, you will have lesser the dosing. Theoretically, you try to understand the epitope, whether it is neutralizing in nature or not. The stability is important of that molecule. Sometimes the antibodies may have very good binding capacity, but they are not stable. The structures they form are not stable. You need to check the cross-reactivity with other similar class of molecules, as well as you need to check the cross-reactivity with certain animal species to understand whether you'll be able to use those molecules for POC, proof of concept studies in animals or not. Once you have at least one or couple of them, which candidates which you have selected, you either do humanization, if required. You do FC engineering, as it was explained in the previous lectures, that you can improve the ADCC, CDC functions. You can also improve the half-life of the molecules by doing mutations in the FC region and improving the affinity towards FcRn binding. And then whether you want to make antibody drug conjugate, you want to make it as just a fragment or bispecific. All these things are then decided. So engineering and optimization step comes. Once your final candidate is selected, you try to characterize it, especially at the level of, again, epitope map, full epitope mapping and kinetic binding profiles. And then you have glycan profiles because they are important for your effector functions. And then you do your POC studies in animals to understand whether your drug is working or not. And once all the studies are done, you then finally try to develop a cell line expressing your antibody or of interest. So how does the workflow of antibody production go, or any recombinant production when it is produced in mammalian cell line go? So you have to do a vector, you need a vector in which you need to insert your genes of interest. Here it can be a light chain and heavy chain in case of monoclonal antibodies. This vector is a mammalian vector, which then you need to transfect into mammalian cell lines like Chinese hamster ovary or other cells. Once the transfection is then done and you have stable clones generated after selection pressure, letting it go through selection pressures and all, you have an scaling up in large scale bioreactors where you take different batches and optimize your process. You optimize your downstream purification process to purify the protein to the highest quality. You then characterize it through a number of different tests, which I'll be coming into, which I'll be showing in the subsequent slides. And then formulating it into a proper formulation so that it remains stable for a longer period of time. Carrying out your preclinical toxicity studies to understand the safety and the dose at which your drug is not going to be toxic. You then, before taking clinical trials, this all happens in an R&D setup. For clinical trials, you need batches to be made in a GMP manufacturing facility. And then you carry out your clinical trials. It can be multiple or single trials based on whether you're developing a novel biologic or a biosimilar. And then once you have all the data, you approach a regulatory to get an approval for launch. Again, we were talking about the cell systems which are being used for expression of therapeutic proteins. So there are a number of them which can be, they can be bacterial cells, they can be non-mammalian cells like yeast, insect cells, and plant cells, as we were discussing. And then you have Chinese hamster, BHK cell line, which have been derived from hamster, human cell lines, and mouse cell lines. Now the difference between all the mammalian cell lines would always be a preferred host for expressing them because they would have a closer property of protein folding and post-translational modifications as humans. Here, this is a pictorial representation of the glycan structures which you see across different species. So what you see in humans is the closest, which is produced by CHO, a Chinese hamster ovary cell line. Though human cell lines like HEC293 and PERSE6 have been used, but they have their own challenges, like you don't get such good levels of productivity here. Secondly, Chinese hamster ovary is the most used cell line in the industry and most preferred because it has been completely characterized currently and its safety has been proven. Many engineerings have been done to improve the productivity and quality of the protein using this cell line. These are the different variants of cell line. I just wanted to bring this to notice as well because, as I said, that we produce monoclonal antibodies in biological systems, and these biological systems vary even at the level of cell lines and then the media, feeds, etc., what you are using them to produce them. In clone this is just a detailed flow of clone generation activities. What I wanted to point out is that after every step of clone generation, or say upstream or downstream, needs its own optimization to be done to have optimal protein production and expression. There are many technologies now which have popped up which help you to screen for high expressing cells. Like for example, there's an instrument called ClonPix2. It's as a semi-solid media where you plate your transfected pool, and then you add an antibody against your monoclonal antibody, which when binds to that monoclonal antibody fluoresces. So high fluorescing cells are picked up. Now it's a regulatory requirement that the cell line which is producing your therapeutic protein of interest has to be monoclonal in nature. That is that it has to come from a single cell. So earlier there were limiting dilutions being done and cells were being observed under microscope, and you used to fish out the single cell clones, but that was very, very laborsome. There was no guarantee whether it's a single cell or not. Now instruments like a clone select imager have come up, which actually show you images while you have plated your single cell into a 96 cell plate. I don't know whether it is clearly visible or not. So what you tend to do is you tend to screen the entire well for the presence of a cell, whether there is a single cell present in it or not. And then across days you monitor the growth, that there is just one single colony being produced from a single cell. So that it gives you the guarantee that your cell is a single cell. You then carry out the productivity experiments to understand the production levels, and you can fish out the high producing clones. You need your clone to be stable for at least some doubling, say 50 or 60 doublings, so that when you scale it up, it remains stable and gives the same level of productivity and quality. And then downstream purification is done. In upstream purification, again, it has its own challenges. There is a certain bioreactor which is usually used at higher scales. Wave bioreactors are also being used, but their scalability is still at very small scale. So once a clone is made, as I said here, it is then taken up by the upstream team, which tries to optimize the process at different levels, like media, feed, sugar optimizations, the stirring optimizations, what oxygenation you are going to give. And there are a number of different parameters which need to be optimized here. So once they are optimized at small scale, they are then taken up at larger scales for taking regulatory batches and supporting the toxicology, preclinical toxicology studies. And after that, they are taken up even at higher scales in GMP facilities. It can be even up to 10,000 liters to 20,000 liters. And there are a number of process controls which you need to keep on monitoring so that you understand that your batch is going well and you will be getting your desired product in terms of productivity as well as quality. So once you start making protein in bulk, purification has to be optimized. For monoclonal antibodies, usually protein A columns are used, which help in eluting out the protein using the FC binding region of your monoclonal antibody. This is followed by a number of different polishing steps where you need to remove the viruses if they are there, or you need to remove the host cell contaminations, its host cell contaminating proteins, DNA, etc. And then finally get a most purified form of protein. So earlier you would have lower purities. As you increase the steps, your purity starts increasing, your yield will start decreasing because you will have the unwanted impurities being removed, and your volume will start decreasing. So this is just a downstream process flow. Now, this is an important diagram which we need to understand. So we have talked about different functions of a monoclonal antibody. But now monoclonal antibodies are prone to degrade or have impurities as time passes, or even they can be generated during your upstream process or during purification process. So these are some impurities which can occur, or these are some modifications which can occur in as a part of monoclonal antibody production, which includes deamidation of asparagine, oxidation of methionine residues, differences in glycosylation, glycation, some conformational changes which can occur, which can be due to the process or formulation, etc. This can occur in variable regions. Similarly, different changes can occur in the FC region of it, like disulfide scrambling can occur, or cleavage can occur, fragmentation can occur, conformational changes, and all those things. So all those things together are going to impact the biological activity of the molecule. Like for example, if you have some changes in the variable region, it can lead to differences in your binding to the target antigen, or changes in your glycosylation in the FC region can lead to your reduced or increased effector functions. And in totality, you need to control them and also characterize this modifications in a monoclonal antibody for its use as a therapeutic protein. Now, these are the various physicochemical tests and biological characterization tests which have to be done to ensure that a monoclonal antibody which is being produced is intended to carry out this activity and is also going to be safe. One is, of course, protein concentration. And also, the elephant shown here is to show that there are different orthogonal methods which have to be used to actually derive or understand the same parameter. So in terms of protein content, in terms of aggregation or size variance, charge isoforms, the glycosylation pattern of it, the sequence of it, the higher order structures, primary, secondary structures, the biological activity of what you are going to produce, as I already said, the residual host cell proteins or DNA, as well as because you're using protein A purification for purifying your product. All these residual entities have to be estimated and analyzed. Secondly, of course, important would be to understand the endotoxin. If some endotoxin has been contaminated, your product is contaminated with some bacterial endotoxin which can happen during the process. So these are the various parameters, and these are actually, you might need to optimize each of this method for each of your product you are doing. And very high-end instruments are used for studying all this physicochemical and biological characterization tests. Now, we talked about there are certain things which can impact the physicochemical property. Like I talked about that oxidation can happen. So what would oxidation do to your biological activity? This is a table of different physicochemical properties and what biological impact it will make on it. For example, oxidation can affect the FcRn binding and pharmacokinetics of the drug. Increase in the mannose content in the glycosylation can increase the clearance and hence reducing the half-life of your drug. And then also it can cause increased immunogenicity in human beings. Decrease in fucose or fucose deficiency can cause enhanced ADCC. In certain cases, you don't need that. In certain cases, it can reduce your ADCC and CDC. So this disulfide scrambling can affect your binding activity. So all these physicochemical parameters are going to lead to some or the other non-wanted biological effects. So it is very important to control them. And because I said that the properties of biological of a monoclonal antibody are very important, so for its biological activity to be maintained, so robustness of your manufacturing process is very important. That every batch of therapeutic protein or monoclonal antibody that you are producing has to be similar, and in case if you are making biosimilar, it has to be similar to the reference product. So I'm just showing you some pictures of the key physicochemical parameters and how they should look like. Like for example, the 3 batches is a reference product, and we are looking at the glycan profile and the different peaks showing different glycoforms. Similarly, this is a pictorial depiction of charge variant profiles where the acidic and basic variants are being shown, and then they have to be similar across the batches and with the reference product. HPSEC is mainly done to understand the impurities, the aggregation profile, which has to be seen across batches, that it has to be robust. The biological potency has to be very close to the reference product and reproducible. The FcRn binding activity is going to be always important because it is going to determine the half-life of your molecule. And then the Fc gamma R3A binding, because it is going to determine your effector functions. So there are this is just, I'm just showing you an overview of certain parameters and the robustness. So what are the major challenges in producing producing monoclonal antibodies or products which are being developed in mammalian cell lines? We already talked about that the cost of monoclonal antibody therapies is pretty high. The reason for that, them being pretty high is the manufacturing challenges with the mammalian cell lines. So when you're going to scale up your process with a cell line which is expressing your therapeutic antibody of interest, it can be challenged by a number of different parameters, like the cells may undergo starvation and stress-induced apoptosis because it doesn't have enough amino acids or enough any component which it requires for its growth and productivity. There is accumulation of metabolites which can occur due to consumption of glucose, which gets finally converted into lactate, which impacts the cell growth and also the quality parameters of the monoclonal antibody. Additionally, ammonia is one of the other metabolite which actually tends to impact your cell growth properties and your quality of your antibody. There are many other things which can happen during your upstream process. And so this actually is causing that the cost of the therapy to be on the higher side. However, in the recent times, because many genetic engineering approaches and process optimizations have taken place, the productivity has moved from less than or equal to 1 gram per liter to up to 7 to 10 grams per liter. And so probably now the therapeutic cost, at least of biosimilars in India, would be coming down and also globally as well. These are the challenges I talked about of a monoclonal antibody or a therapeutic protein. Now, if we talk about more complex formats being made like bispecific and ADCs, it will tend to have its own challenges. Like, for example, a bispecific antibodies, this is the format which is desired, where you have 2 different light chains and 2 different heavy chains to bind to 2 different targets. So when you are going to produce this in a Chinese hamster ovary cell line, it can tend to give many different isoforms. And so there are methods and genetic engineering approaches which have been made to control this. So ultimately this will lead to reduced yields and unwanted antibody impurities, which would then increase the cost of production of bispecific antibodies. Similarly, anti-drug antibody conjugates, as we saw, that it's a mixture of antibody which is linked to a payload or a chemotherapeutic drug. So when you are carrying out reactions, they can be this chemistry can be non-specific, and it can lead to a large number of different variants being produced. So controlling the DAR variants is a very important challenge and a great challenge in making anti-drug antibody. If you have very high DAR ratio, it can lead to safety issues, and if you have very low DAR ratio, then it can lead to efficacy issues. So this balance is very, very important. Now coming to the formulation, we saw everything right from cell line generation to purification to physicochemical characterization. Formulation, usually monoclonal antibody drugs have to be given at very high doses, and hence they are given in form of IV because it is very difficult to concentrate them, and concentrated formulations may not be very nicely absorbed if they are given in a subcut form. So in the recent times, monoclonal antibodies have been developed in a subcut formulation using a protein called hyaluronidase. Now this hyaluronidase protein, when co-formulated with a monoclonal antibody, what it helps is that it goes and degrades a hyaluronan which is present on the surface of your skin, and it creates space for big molecules like monoclonal antibodies to easily enter into your bloodstream. So this is one area again which is novel, and this reduces your time of injection. IVs, we all know it has a long time period. It can be given up to even up to 2 hours, and high volumes have to be given. But when in the subcut formulation space, this will be an ease for patient to deliver the same. Secondly, Sanjeev sir had talked about in the morning that once your drug is made, when you do your clinical trial, a number of different assessments have to be done, especially PK and immunogenicity have to be studied. And it itself is a big challenge. I'll not go into the details of it, looking at the time. I guess I've run out of time. But you have to develop assays which are sensitive enough to pick up enough amount of drug when given into a human body or into animals, so that you can have an entire PK profile which can be properly studied. Also, immunogenicity assays have to be done because any protein which is given from outside is going to be foreign for that patient or human being. So there are different methods which have to be developed, not going into detail. But yes, it's a challenge to develop these assays, and you have to work for months together to get correct type of PK and immunogenicity assays. One thing I just wanted to point out, we have been talking about development of therapeutics during the pandemic times and COVID, etc. So usually if you see the time from developing a novel biologic to filing an IND after doing tox studies from a global perspective is 12 months. While with the COVID monoclonal antibodies, we could achieve it just in 6 months. So rules are changing, people are understanding the space better, and so antibodies are being discovered at a much faster pace. And in fact, also the monoclonal antibodies against COVID were actually released in a they were launched in quite a short time, around 2 years. Otherwise, the development after clinical trial takes around 5 to 8 years. So this is something, a good point that in pandemic situations as well, we'll be able to cater to the population and help them out. So this is my last slide. I talked about the development of antibodies and what are the challenges with it. Just to bring to your notice that what antibodies have already been made by Zydus and they are in the market. So adalimumab biosimilar was the first biosimilar in the world to be launched by Zydus in December 2014. Vivitra is another monoclonal antibody. It's a biosimilar of trastuzumab, which we had launched in 2015. Brixta is a biosimilar of bevacizumab launched in 2017. TwinRab is a cocktail of monoclonal antibodies which we developed in collaboration with WHO. It is basically an antibody cocktail which has to be given before vaccination. The importance of it being that vaccine takes its own time to produce antibodies and neutralize the virus. So you need something which can neutralize the virus ASAP. So you give this antibody so that the virus neutralization occurs as soon as possible, and you are able to downregulate the further damage. We were again the world's first company to launch a biosimilar of antibody drug conjugate, which is a biosimilar of Kadcyla or trastuzumab emtansine, which is Ujvira. In addition to this year itself, we have completed clinical trials for 2 additional mAbs, and probably will be getting approval for the same to launch in this year. So this is what the monoclonal antibodies with Zydus has been working on. There are a number of them in novel biologics in preclinical or discovery phases, as well as in the, there are biosimilars also in earlier discovery phases. Thank you so much.
The floor is open for questions for Dr. Parikh.
Bispecific antibody. So as you mentioned, if they bring the proximity to the T cells and maybe cancer cells. So one part of the antibody has the immune counterpart, another is a cancer counterpart. But I'm just asking a biological question, curiosity. Say there is not a chance comes like the immune cell is not in the proximity, you have cancer cells. And if it binds with the one receptors, can that be internalized with the other counterpart?
Internalize with the
It's a receptor biology, right?
Yeah.
The antibody is going.
So all antibodies will not be internalized after target binding, right?
Just asking.
No, that is not the case. Not all antibodies bind to the target and get internalized.
No, I'm not talking about all antibodies. So
in this case, the example which I showed, one was, I guess, CD20, which is expressed on T cells, and the other is, CD20, which is expressed on tumor cells, and the other is the CD3, which is expressed on T cells.
Right. So the one that is expressed on tumor cell and the antibody specific to that receptor binds. Okay. Right. And the immune part is not in the proximity.
Yeah,
Forget
it can happen.
One.
It can happen.
And that receptor binding can internalize that combo?
I don't think there have been reports of it, as far as my knowledge goes. But yes, sometimes it finds difficulty in finding if it is not in proximity. But to bring it in proximity only, you have something which have 2 different affinities. Sometimes it is like that you have a higher affinity for some target which is expressed on tumor cells, while you might have lower affinity for the T cells. So it is a bridge, and it is sort of a balance which has to be maintained. And that is how you try to bring 2 cells together and have its function done. It can also be a CD19, CD20 bispecific, where you have 2 targets on the cancer cell itself. So it can bind on the same cancer cell or a different cancer cell and lead to its death.
So another thing, you have given some examples of antibody drug conjugates. We have long back, and few groups also found that this antibodies really is a good targeting when you use in the in vitro, while we use, especially in the nanoparticles, drug conjugates, as a targeting agent, these antibodies. But when you use the same conjugates for in vivo, that didn't translate that well.
That would happen with many, even IL-2 example as Madam gave. I mean, in the space of biotherapeutics, you'll have to check what happens in vitro and what happens in vivo.
So the one you were mentioning, they had shown promises.
Yeah.
There
are multiple cancer cell lines which are used to test before taking it into the in vivo studies or taking it into humans.
So they translated.
So they did, they translated. The approved ones did translate, yes.
Okay, that's all.
There was a question here in the middle. Yeah, please go ahead. Please go ahead.
Hello, ma'am. I had question that what is the difference between a biosimilar and a novel drug? Like if they are clinically same, if their bioactivity is same, then at what point they differ?
No, so a biosimilar is a version or a copy of already existing product. For example, erythropoietin. It's already existing. Its sequence is known. The same sequence you use and produce it in your lab. That is called a biosimilar. When I see a novel biologic, it is something which is new. It is not there in the market or not being used for treating patients. That is called a novel biologic. It has not been yet tested and approved.
Okay, so novel drug is not in the market yet. It is
Right.
just in the laboratory.
Correct.
Okay. So people patent those novel drugs?
Yes.
So we
Anything
make
new.
biosimilar after the patent is over?
Right, yeah. So that is a challenge, and that is why India is not able to make all biosimilars. Otherwise, we have the ability to make a copy of each and every existing drug. But we have to take care of the patents.
Okay.
So as the patent expiry nears, companies start developing those drugs.
Okay, and generic drug is another different category.
Generic is a terminology which is used for small molecules, chemical molecules. Just I gave an example of paracetamol. You have paracetamol from many different companies, or even statin drugs, or many drugs you can say. They are being marketed by many different companies, right?
So can we say that generic drug is a biosimilar?
No, the scientific definition is, that is why I tried to explain. The generic drug is given because you can make almost an exact copy of it. These are regulatory definitions which are given. And biosimilar you say because it might not be an exact copy, because of the biological systems being involved which differ in producing that particular molecule of interest.
We will go to the next
Okay,
question. There was a question at the back. Was there a question at the back? Yeah, please go ahead.
You mentioned earlier that the antibody drug ratio ranges from 3 to 5. So on what basis it is decided?
So it is decided based on, it has to be a balance of toxicity. It is, as I even earlier said, it has to be a balance of efficacy and toxicity, right? These are chemotherapeutic drugs. So at particular ratios, they can tend to cause toxicities. So 3 to 5 is something which I gave an average of what molecules are in the market. Now there are even more, there are modifications being made to the payload itself so that you can have higher DAR ratios or the payload which is attached, like Durextican. It can attach up to 7 to 8 molecules and yet not be toxic. Right, there are also linker chemistries being played so that they go and just get released into the tumor microenvironment, and they'll be very safe. More drug will be getting released into that environment at a lesser dosage, and you have more efficacy and less toxicity. So all those research is going on in that area.
Any further question? Last question from anybody? Okay, let's thank Dr. Parikh for an excellent talk.
Thank you, ma'am, for your insightful talk. I request Dr. Sehgal to felicitate Dr. Aashini Parikh. Yeah, I request all of you to proceed for a tea break and report back by 5:30. Thank you.